In this video clip, we'll continue to talk about some of antimicrobial drugs and how they actually work. So part 2 for interactions between drugs and microbes. All right, so we talked about antibacterial drugs in the last video clip that impact bacterial cell walls and bacterial ribosomes. Okay, so in this video clip, we will talk about some drugs that are not necessarily antibacterial. So there are drugs that disrupt cytoplasmic membranes of microorganisms and kill them. So basically they poke holes in the membrane and that causes the microorganism to die. There aren't very many of these though, because it's difficult to come up with drugs that will punch holes in the membranes of a microbe and not also do the same to our cells. So there are some toxicity problems with these types of drugs for us in a lot of cases. Now, one example of a good antimicrobial drug that disrupts cytoplasmic membranes is called amphotericin B. This is an antifungal drug. So you wouldn't take this for a bacterial infection. This would be for fungal infections, especially fungal infections deep inside the body, systemic types of fungal infections that are occurring across many body systems. And so how this is what the chemical structure of amphotericin B looks like. And over here we've got a little chunk of a cell membrane from a fungus. And what this drug does is it attaches to, there are special lipids in fungal cell membranes that are called ergosterol. And this thing that kind of looks like a purple key there. And that's closely related to cholesterol. We have cholesterol in our cell membranes. Fungi have ergosterol. So this drug attaches to ergosterol and winds up creating pores in the fungal cell membranes, and that causes the poor little cells to die. They lyse or burst. Now this drug can be, you have to be careful with it, because if you take it at too high of a dose or for too long of a period of time, it can be toxic, because amphotericin B also weakly attaches to cholesterol that we have in our cell membranes. So if you take too much of the drug, that's going to increase the likelihood that that drug is going to wind up poking holes in your cell membranes as well. So that's would not be a good thing. Amphotericin B does not work against bacteria. Bacterial cytoplasmic membranes don't have ergosterol in them. Another one is polymyxin. Now this is a drug that disrupts bacterial cytoplasmic membranes. It's particularly used with gram-negative bacteria, especially Pseudomonas aeruginosa, which is a common microbe out in the environment and generally doesn't cause problems with patients who don't have or aren't compromised in some way. But if you are immune compromised, or if you are a burn victim, so you've lost that skin barrier protection that you guys learn about when you take Biology 201, you are at a high risk of Pseudomonas infection, because Pseudomonas is out there everywhere. But unfortunately, we don't have a lot of drugs. In some cases, polymyxin can be the only drug that's useful for some of these infections. And unfortunately, polymyxin is toxic to the kidneys. You have those delicate structures in the kidneys that filter the blood and create urine, and they're especially susceptible to damage from polymyxin. So there are some cases where you can have a patient who is infected with some organism that's resistant to almost every antibacterial drug out there except polymyxin, and your choice is the patient is going to die from the bacterial infection, or you're going to give them polymyxin and they're going to wind up with kidney damage from taking that drug. So sometimes, because of all this antimicrobial resistance that's developed out there, we're already facing some tough choices with patients at times, which is kind of sad. All right, there are drugs that inhibit other metabolic pathways. They home in on something very specific. There's one called atovaquone that interferes with the electron transport chain. Remember that from cellular respiration and protozoans and fungi. But apparently doesn't interfere with it enough in our cells to cause us problems if you take the drug at safe doses and for safe periods of time. The sulfonamide drugs, or sulfa drugs, these were the first really effective and useful antibacterial drugs. So they were discovered a little bit before penicillin. And so they're kind of interesting in how they work. So here's an example. Here's sulfamethoxazole. And so it has this chemical structure that very closely resembles the chemical structure of a compound in bacteria called PABA, P-A-B-A. Now that's an abbreviation for a very long organic chemistry name. But this chemical compound in bacteria called PABA is converted to folic acid. And folic acid is a coenzyme. That's a little, remember, those are chemicals that enzymes need to help carry out their chemical reactions. Folic acid is used for nucleotide synthesis. So without folic acid, you can't build your nucleotides properly in bacteria. Or in our cells as well. Humans says we need folic acid. Where have you heard about people taking folic acid supplements before? And if you have heard of it, it's because they're given to pregnant women, because folic acid is being used in great abundance by the developing embryo or fetus in the womb, because there's a whole lot of nucleotide synthesis going on in the womb. So folic acid, by giving that as a supplement, it helps make sure that that developing embryo and fetus can build all of the nucleotides it needs for its DNA and its RNA. All right, so how these sulfonamides work. So there are enzymes that convert PABA to folic acid. These drugs, what they do is they inhibit those enzymes because they're mimicking PABA. The enzymes attach to the drug instead of PABA, and now PABA doesn't get converted to folic acid. All right, so you have limited numbers of nucleotides, you can't build your DNA and your RNA, and the cell winds up dying. So these are good because our cells, we have to take in folic acid in our diets. We can't build, we don't convert, our cells don't convert PABA to folic acid. So since these drugs are very specific for that metabolic pathway, those chemical reactions, they're useful for killing bacteria without harming our cells. All right, another group of drugs are the quinolones or the fluoroquinolones. So now these are antibacterials as well, and they're very specific about what they interfere with. They inhibit an enzyme called DNA gyrase, that's an enzyme that is necessary for unwinding. You know, we've talked about how DNA was very compact, or in Chapter 8 we talked about how DNA is very tightly compacted down, all wadded up and folded up so that it can fit inside cells. And so these drugs interfere with an enzyme called DNA gyrase that helps relax the DNA so that it can be replicated when it's time for it to be copied. So these drugs interfere with that enzyme, and by doing that, it helps prevent bacterial DNA replication. And luckily, these drugs don't interfere with our DNA replication. You've probably heard of Cipro before. Cipro is an abbreviation for ciprofloxacin, often used for treating UTIs, urinary tract infections. That's an older drug. There are some newer ones in this fluoroquinolone category, like gadafloxacin, gemafloxacin, that work in a similar way. So those are antibacterials as well. A number of drugs out there interfere with nucleic acid synthesis in other ways. So we just talked about how the sulfonamide drugs prevent bacteria from making the folic acid that they need to build DNA and RNA nucleotides. But there are other drugs that interfere with nucleic acid synthesis as well. Many of these are what we call nucleoside analogs. So if you guys remember, a nucleotide is the nitrogen-containing base, like adenine, guanine, thymine, or cytosine. And then that is connected to a sugar, either ribose or deoxyribose, DNA or RNA. And then that's connected to a phosphate. Those are your three components of a nucleotide. A nucleoside is just the nitrogen-containing base and the sugar, so there's no phosphate attached to it. And these nucleosides can be taken up by cells, and then once they go inside a cell, the phosphate gets attached, and now you have a nucleotide. So what nucleoside analogs are, these are drugs that mimic naturally occurring nucleosides. So you see those here in the center, like adenosine would be an adenine nitrogen-containing base attached to the sugar. Guanosine is a guanine nitrogen-containing base attached to a sugar. Okay, so those can be taken up by cells, a phosphate gets attached, and now you have a nucleotide. Now what you're seeing surrounding those, these are various nucleoside analog drugs, so they are mimicking the naturally occurring nucleosides. And so what happens is the enzymes that would use these to make nucleotides and then use these for DNA or RNA production will instead use these analogs, these drugs. And when these drug forms, when they get incorporated into DNA or RNA, whichever they're being used for, it stops the chain. So if you incorporate one of these drugs in a strand of DNA that's being built, that's it. The chain terminates. You're not going to be able to add any additional nucleotides. Or if you're doing transcription of RNA and you incorporate one of these, transcription stops. And so these can be lethal for the microorganisms that are being treated with these types of drugs. Some of these may have familiar names, like acyclovir is mimicking G nucleosides, guanosine. Ganciclovir, those are pretty commonly used. Those are antiviral type drugs. This one, AZT, stands for azithromycin. It is an analog. It's mimicking the thymine nucleoside. Okay, and you may have heard of this one before because this was the first widely used drug for HIV infections. Because this drug will inhibit HIV reverse transcriptase, so it helps prevent the conversion of the RNA genome of HIV to the DNA version. If you watched the movie Dallas Buyers Club that came out a couple of years ago, and AZT was the drug that they were selling on the black market back in the 1980s in Dallas to the AIDS patients who were desperate for drugs to teach their illness. So a lot of these are antiviral drugs. This one, dideoxycytosine, DDC, is actually an anti-cancer drug. So cancer cells do a lot of DNA replication because they're dividing very, very quickly. So drugs that interfere with DNA synthesis will typically kill cancer cells. Unfortunately, they're also going to be toxic for other cells in your body that are undergoing cell division as well. Another drug, rifampin, is a drug that inhibits prokaryotic RNA polymerase, so it's going to mess up prokaryotic transcription, but hopefully not eukaryotic transcription. So that's another example of a drug that can inhibit nucleic acid synthesis. A number of these you have to be careful with. A lot of them are antivirals, but they can also interfere with processes in human host cells as well. All right, this is a table from your textbook, which is showing you some examples of spectrum of drug activity, narrow spectrum versus broad spectrum, and there are some different categories of microorganisms up here. These are all bacteria. Mycobacteria, gram-negative, gram-positive, chlamydias, I'm sure you've heard of chlamydia before, sexually transmitted disease. Rickettsias are little teeny tiny bacteria that live inside host cells, kind of like viruses do. And so, for example, the tetracyclines here, which interfere with protein synthesis in bacteria, that's a broad spectrum antibacterial because those drugs impact gram-negative bacteria, gram-positive, chlamydias, and ricketsias. Something like tobramycin or polymyxin, those are much more restricted. Tobramycin is used for mycobacteria and some gram-negative bacteria. Polymyxin is used exclusively on some kinds of gram-negative bacteria. So you've got broad spectrum drugs, you've got narrow spectrum drugs, and you've got drugs that have spectra in between the two. All right, kind of a downside with broad spectrum drugs. If you're taking something, now sometimes you go to the doctor, and if you're pretty seriously ill and he or she thinks you've got a bacterial infection, and they don't have time to figure out exactly what's causing the infection to kind of narrow down a good drug, they're probably going to prescribe you a broad spectrum antibacterial that would kill a lot of different types of bacteria. A problem with that is, and we've talked about this before, that's going to kill a lot of your normal microbiota, those friendly bacteria that live on and inside your body as well. And when that happens, it opens the door for resistant microbes to overgrow. So we've talked about how Clostridium difficile can do that in the colon. Candida albicans, remember that? That's yeast, single-celled fungus. Fungi aren't susceptible to antibacterial drugs, so if you've knocked out a lot of the bacteria, the Candida albicans, yeast that naturally live on or inside the body, have the opportunity to overgrow because they don't have as much competition for nutrients. It's kind of like they're at a big breakfast buffet and nobody else is in the restaurant. So Candida tends to overgrow along the vaginal mucosal membrane. So it's not unusual for some women to develop yeast infections following taking a broad spectrum antibacterial drug. Or little kids especially sometimes get thrush. That's an overgrowth of Candida albicans yeast in the mouth. And sometimes that can follow treatment with a broad spectrum antibacterial drug as well. Think about biofilms again. Biofilms, we're appreciating more and more that if microorganisms are present inside of the protective covering of a biofilm, it's harder to treat the infection because the drug just can't get to them. It's got to go inside the bacteria to interfere with protein synthesis or cell wall synthesis or production of folic acid or whatever that drug is designed to interfere with. You got to go inside the cells to make that happen. Okay, this is a table from your textbook which gives you some additional examples of antiviral drugs. I think a couple of these I just mentioned over on the nucleoside analog location. We don't yet have nearly as many antiviral drugs as we do for bacterial infections. And again, it's just hard to come up with drugs that interfere with something that a virus does but won't cause problems for the host cell that they're inside, because viruses replicate inside host cells. But kind of the targets are, we're coming up with drugs that block virus entry. So you block the virus from attaching to its receptor, or you block the uncoding of the genome once the virus has entered. Or you screw up nucleic acid synthesis, DNA or RNA genome production. And some drugs interfere with the assembly of viruses or the release, like let's say budding. That's something that can be interfered with as well. So take a look at this table just kind of for a general overview of some of the antiviral drugs that are currently out there and how they actually work. All right, now one thing we're not really going to do, your textbook gives some overviews of drugs that are available for treating fungal, protozoan, and helminthic infections, and I'm having you read that information. But other than the amphotericin B that I just mentioned earlier, I'm not going to ask you questions about those particular types of drugs. So again, be sure you're using your study guide learning objectives very carefully while you review this material, because I've cut some information from both Chapter 9 and Chapter 10 because we just simply don't have time to go over all of the information in all of these chapters in a one-semester microbiology course. All right, so that's just kind of an overview of some antimicrobial drugs, and again, we mainly focused on antibacterial ones. As we move into this fourth video clip, now we're going to talk more about antimicrobial resistance. How can a microbe resist a particular drug? And then how are they changing? What's going on that's causing more and more microorganisms to become resistant to the drugs that we're using? This is a huge topic for people who are going into healthcare, so that'll be the topic of our fourth video clip for Chapter 10.